So my name is Heather Shappell. I'm in the Department of Biostatistics and Data Science at Wake Forest University. I guess I stand between you and lunch now. But I ended up not including a lot of equations in my talk. So I don't know if that's good or bad, but it might be good for the pre-lunchtime talk when you're a little tired and hungry. So as a quick overview of my talk, I'll go through a little bit of background on fMRI and brain networks. I'm assuming most people here are probably semi-familiar with it, but I know there are a variety of backgrounds here, so I want to at least touch on it a little bit. And then I'll talk about a hidden semi-Markov model approach that I'm proposing for dynamic network analysis. And then I will discuss an application to an ADHD study. And if we have time, I'll talk about some extensions that I'm working on. Okay, so as we all know, brain activity is constantly fluctuating as we engage in different tasks and activities. And the brain has many specialized areas, right, that have different roles but all work together. So a brain area for language processing, for memory, for helping you with motor tasks. But then these are ultimately all working together. And fMRI, functional magnetic resonance imaging, helps measure and map that brain activity in a safe and non-invasive way. So because we have these different brain regions and they're all sort of working together, it makes sense to think of it as a network representation where different areas of the brain are the different nodes in the network, and then edges represent areas that are either actually physically connected, like in an anatomical network, or areas that are sort of communicating with each other and activated and deactivated at similar times, and that would be more so of a functional network. So I am a statistician and not a neuroscientist. So this is my very naive, kind of big picture understanding of fMRI. So I wasn't sure if everybody was familiar with fMRI, and neuroscientists in the room might be laughing at this explanation, but this is how I understand it, that neurons do not have internal reserves of energy in the form of sugar and oxygen, and blood releases oxygen to active neurons at a greater rate compared to inactive neurons. And then this causes a change in oxygenated or deoxygenated blood that can be detected by the scanner. So basically detected on their basis of different magnetic susceptibility. So if we were actually going to construct a functional brain network, this is kind of a big picture representation of how you might go about doing this. We start with A, where you actually collect the fMRI time series data throughout the time that somebody is in the scanner, right? And we get what are called BOLD signals, blood oxygen level dependent signals. So you kind of have a snapshot of this over time throughout the time they're in the scanner. So you have this fMRI time series data. And then a lot of pre-processing goes into this. And pre-processing is not something that I particularly work on, but I know there's a lot of work that has gone into, for example, like detecting motion artifacts and other types of artifacts and pulling out the real signal and warping people's brains onto a similar brain space so that you can compare across people or groups. So there's a lot of pre-processing steps that go in. And then you can sort of parcellate the brain, and there's different ways of doing that. We won't get into that into this talk, but you can parcellate the brain into different segments or regions, and then extract those BOLD signals at each of those regions, and you have that for the time that somebody is in the scanner, right? So if you parcellate the brain into 400 regions, you have that time series data of BOLD signal at each of those 400 regions. So you have that time course extraction step, and then you can create what is called, it's like a weighted network actually, this correlation matrix, right? Where you correlate each pair of the time series data at so for every pair of those regions, you have a correlation coefficient. People use different ways of doing this. We typically use Pearson correlation in my work. So correlation coefficient, and this correlation matrix here, so you can think of it as, say you have 100 regions, 100 by 100, And you go down to region 2 and you go across to region 3 and you look at the little square, and that would represent the correlation between those 2 regions. So in this matrix, red would be a higher correlation, and perhaps maybe blue would be a stronger negative correlation, and then you have some greens in the middle for moderate correlations. So pairs that have a higher correlation value, we would typically think of as being more synchronized in their activity. And if we were going to actually create a binarized network, we could threshold and say, anything above a correlation value of maybe 0.5, we'll define an edge between those regions, and anything below, we won't define an edge, if you were actually creating this binarized connectivity matrix. Or oftentimes, you can just kind of leave it as this and look at it as a weighted network. So if you are binarizing, then you can kind of create a functional brain network from there, and then you can perform all types of graph theory analyses on this, where you look at things like modularity and clustering and density, which some of those statistics also exist for weighted networks as well. So that was a little bit of background. Really the prevalence, as you know, because you're sitting here, the prevalence of networks in neuroscience has grown tremendously over the past decade. And this is evidenced by big projects such as the Human Connectome Project, the 1000 Functional Connectomes Project, UK Biobank, where they're collecting these massive amounts of brain data and really getting to the point where we're doing a pretty good job of mapping the human brain. So my work has focused on functional connectivity, as I was just talking about, as opposed to structural connectivity where there's an actual physical connection. Functional connectivity, we're looking at the correlation or the association between those bold signals over time that someone is in a scanner. And if they're highly correlated, we're saying, yes, they're probably working together in some way to carry out tasks in the brain. And the neat thing about functional connectivity is it's already shown to form specific patterns that are reproducible across individuals and predictive of individual variations in cognition. So there have been several techniques that have been proposed for doing this. Now, so far, when I've described this, I sort of described it as a static network where, okay, you take the whole time series that someone is in a scanner, right, and you create one network. And that's what people did initially. They just got one static network for that whole length of time they were in the scanner. But then we started thinking of, well, people are probably transitioning in and out of multiple brain states, right, or brain networks. It doesn't really make sense to think that they're in one just kind of average network throughout the whole time. So can we figure out multiple networks that they're in and out of, and when they're switching to different networks? So this is dynamic connectivity. And methods have been proposed to do this, such as the very, very common sliding window analysis, where basically you set a window length, like maybe 50, and you get the first 50 time points that they're in the scanner, right? And you calculate a correlation matrix for that, and that's a network. And then you shift it over by 1 or however many, and you calculate another correlation matrix, and you shift it again, and another. So you get this whole sequence here of covariance or correlation matrices. And then a lot of times people will perform like a k-means clustering type algorithm where they'll say, okay, now cluster these into like 6 brain network states. And they'll get those 6 or however many, and then they'll be able to say, okay, yes, this individual was in the first one for the first 10 TRs or 10 time points in the scanner, then they switched to this other one, and then they went back to the first one, and so on. But this approach is common, it's very easy to perform, but it can be a little problematic because, one, what do you set your window length to be, right? If you set it to be kind of too short, then your estimates might be a little unstable. If you go too long, you're going to miss some of the quicker changes in connectivity, right? Some of the changes will kind of get washed out. So, and that's the other thing, is this approach does kind of miss some of the quicker changes. Like if someone is bouncing back and forth pretty rapidly between multiple networks, this method has trouble detecting that. So I was looking into other approaches for this, and I saw that one or two people, this was a few years ago now, had started using standard hidden Markov models for this type of analysis, where basically that top layer that you see there, right? This is what you don't know. So this is the brain network state, we call it, that someone is in, but you don't know what it is. And what you are observing are those bold signals. So at each time point, we observe the Ys, we observe the bold signals there at each of our regions that we've parcellated the brain into. And those states that we can't observe, that hidden layer, gives rise to those bold signals, right? But we don't know what those states actually are. That's what we want to estimate. So those states represent unique brain networks of distinct activity and functional connectivity that repeat at different points in time. So maybe someone started in the blue state, switched to red, stayed in red, went back to blue, right? So we want to be able to estimate those. So basically, we assume that the Ys follow a multivariate Gaussian distribution dependent on what state they're in, right? So each state will have its own mean and covariance matrix. And you can think of the covariance matrix as what's defining that network, right? Because you can get a correlation matrix from the covariance matrix, and that's the networks that I basically showed here on this slide. So we want to estimate those for each state. So that's like estimating our network for each state then. So the Ys depend on the states, right? They follow a multivariate Gaussian distribution with the mean and covariance that depend on the state. That's this first part. If we were to write out this complete data log likelihood, that is this first part here. And then the second part here are the transition probabilities of going from one state to another. So going from blue to red, or from red to yellow, right? So we want to estimate those transition probabilities. And then the last part here is just the probability of what state you start in. So there are some advantages of this approach. You can actually estimate large data sets here, and it's relatively computationally inexpensive. So basically, you can fit this on a lot of people and get a set of states, right? Get a set of covariance matrices, so a set of networks. You can get the transition probabilities. You can get the initial state probabilities. But then what is neat is then, based on those estimates that you get, we use the EM algorithm to estimate those. Based on the estimates that you get, you can go back and use what's called the Viterbi algorithm to say, given these estimates, given this particular subject's time series data, what state are they in at each time point? Like, given that set of states you estimated. So basically, the states will be estimated at the group level, but then the individuality is respected in when the state is active for each person. Also, the nice thing is the method is likelihood-based, which allows for model selection techniques such as what is the most optimal number of states for this group of people? Like, is it 6, is it 7, 8, whatever. And the model, unlike the sliding window approach, is able to capture quicker changes in brain activity. But the main limitation that I did not like about this approach is that the sojourn time, and by sojourn time I mean the amount of time that you dwell in a state, that you spend before you jump out to another state, is assumed to be geometrically distributed. And this is kind of implicit here in this model setup, right? So if you think about having you're estimating your transition probability matrix, the amount of time that you spend in state 1 before switching to another state, right? That's exactly a geometric distribution. A geometric distribution is, number of times before the first occurrence of something. So this ends up, if you think of a geometric distribution, it's always decreasing, right? It's like this. And so what this is saying is you have a higher probability of only spending 1 or 2 or 3 consecutive time points in a state before you jump out to another state. And what if that's not true? Like in some tasks, for example, you might remain in a state for 10 TRs on average while you're performing the task before you jump to another task. And so this is sort of biasing the results to encourage kind of quicker state switches. It's having the model estimate quicker state switches than are actually probably happening. So we proposed using a hidden semi-Markov model to estimate our states and our transition probabilities and our state sequences for each person. So I've kind of flipped here. Now I have the observed data up top. Our Ys, again, are the observed data, right? Each is a vector containing the bold signals at p different nodes. And then you have this latent state sequence that you are trying to estimate that you don't know. These are the networks that somebody is in, and they give rise to the bold signals. So now we still have, to estimate, these red arrows are denote the transition probabilities. We still have to estimate those transition probabilities. But now we can put a distribution and that's not geometric to model the actual time that someone is spending in a state. So maybe we fit a Poisson distribution, for example. And we use that to determine the probability of only staying 2 time points in a state. And then 3 time points in a state. And then only 1 time point, right? So you can fit Poisson, gamma, normal, whatever you want. You don't have to assume that it is geometric. So now, this will be directly in the model. We're going to explicitly define a Sojourn distribution, and we'll be able to estimate those parameters in the model, and it doesn't have to be geometric. So before I show you the likelihood of that, let's denote each participant's fMRI time series data by yi1 through yit, where each p-dimensional vector yit contains the bold measurements of the p ROIs. Those are regions of interest in the brain that we parcellated the brain into, at the tth time point for the ith participant. Again, we assume that each yit follows a multivariate Gaussian distribution, where the mean and the covariance are dependent on the current unknown network state at time point t for the ith participant. So you can write this complete data, and by complete data I mean, if you actually knew the true network sequence, that hidden layer, if you knew that and you have the observed bold signals, you can write it as this. This first part is pretty much what we had before, right? The bold signals given the underlying states, that follows the multivariate Gaussian distribution. And then this next part here is what changes. Now we actually separate basically this into segments, where each kind of segment is where someone is in the same state. So there's going to be R segments of those. And then we have the probability of transitioning once they do transition to another. That's here. And then we have the actual Sojourn distribution right here. And again, you can make this Poisson, gamma, whatever you want. So that's explicitly in the model now. It's not tied up in that transition probability matrix. And then this last part here is just for whatever state you start in, the probability of starting in a particular state, and then remaining in that state for a certain amount of time. That's the Sojourn distribution. So U sub r is the number of consecutive time points that you spent in the state, and then here is the Sojourn density for that state. So I don't have all the details here. The tricky part is actually estimating all of this stuff. And as I said earlier, we use the EM algorithm to do that. If anybody wants to chat about that later, I'm happy to do so. But originally, I had some equations in here for the expectation step and the maximization step. And then I figured that's not as interesting. So we wanted to test this on, we wanted to kind of do a sanity check on this experiment that some collaborators already had data for. They called it the Anxiety-Inducing Experiment. Where basically they had 23 participants perform this anxiety-inducing task while in the scanner. So this is the paradigm here. They started off with just kind of a fixation cross or something that they were staring at in the scanner. And then they were shown a slide where it said, you're going to be given 2 minutes to prepare a 7-minute speech. And they were given a speech topic. And then they had time to prepare this speech. And then after they spent 2 minutes panicking and preparing the speech, then they were shown a slide that said, actually, you were randomly chosen to not have to give the speech, because they told them they were going to have to give the speech to a panel of like judges or something. So then they were saying, actually, you were randomly chosen, you're not going to have to give the speech. And then they ended with just a fixation period. When I was giving this part of this talk as a job talk, I tried to make a joke in the middle, and I said, you know, it's funny, no one told me today that I wasn't going to have to give my speech today. I don't know if they really found that funny. So, again, this is just sort of a sanity check. So we only used 4 regions of interest in the brain. This is not supposed to be anything groundbreaking here. We just wanted to see if our hidden semi-Markov model can actually pick up on the switching at the appropriate times. Like we were hoping to see non-anxiety, and then all of a sudden anxiety state, and then non-anxiety again. So we used 4 regions of interest, and then heart rate as sort of like a dummy region. And the 4 regions were chosen based on the fact that they showed a significant relationship with heart rate in another data set. So the regions were actually 2 different subsections of the ventral media prefrontal cortex, which is red and blue in these figures, the dorsolateral prefrontal cortex, which is purple, and the ventral striatum green. And then heart rate is this little pink dummy region down here. So our model estimated 2 different states. I'm calling this one the low anxiety state and this one the high anxiety state, where the high anxiety state basically has a connection between all the different regions, all 4 of them, and heart rate. So here is a heat map. After we fit the model, we then went back for each subject and we estimated, given the states that the model estimated, given the transition probabilities, given all that, using that Viterbi algorithm I mentioned, which state is each subject in at each time point in the scanner. And this heat map here, the darker red means the more subjects fell in that state. So time is down here, and we have state here on the y-axis. So you can see most people, because this is red, most people start in state 2. State 2 was actually, I should have split these. State 2 is the low anxiety state. So most people start in the low anxiety state. Yellow means very few people started in this higher anxiety state, but some people did. Some people are nervous right when they get in the scanner. And then here where I have the black vertical bars, that is where they had the slide that said you're going to have to give the speech. And so you can see that now we start to get a little bit more red up here. So people start transitioning into that higher anxiety state. And then they're in that high anxiety state. Almost everyone is, and everyone is for a big chunk of time here while they're preparing the speech. And then they start to drift out of it again. And then when they're shown that slide saying you're not going to have to give it, then most people go back to the low anxiety state. So the model did seem to pick up on this kind of very simple 2-state sanity check that we did. And then, because we are interested in Sojourn distributions, right, we're saying that this approach can accurately estimate Sojourn distributions, we estimated the Sojourn distribution from the model for each state. So we fit a Poisson Sojourn distribution. So state 1 here is black. This is the Poisson distribution that was fit. And it seems pretty accurate. I mean, it's saying that they spend around 60-ish time points in state 1, which is, pretty accurate here. And then a little bit less in state 2, which is the high anxiety state. It's shifted a little bit. So they spent, a little bit less time, a little bit consecutive time in that state before switching out of it. And that's probably true too. I mean, after people prepared their speech, they probably got a little bit less nervous and jumped back out. So this seems to make sense. Now, just to kind of compare, I then fit the standard hidden Markov model on this, and it gave us geometric distributions, right? And this is what it's saying if we were to fit the standard hidden Markov model, would be the Sojourn distribution for each state. And again, this isn't really accurate. It's saying there's a higher probability for only spending a couple time points before switching out of it, which is not true. At least we don't think it should be true. So I didn't end up showing simulation results here. We actually used the SimTB toolbox. I don't know if anybody else has used that. Vince Calhoun's group has developed that to simulate fMRI data. And then we tried the sliding window approach. We tried, so let me take a step back. We simulated fMRI data, and then we simulated, people, basically dynamic connectivity, different states that individuals would transition in and out of. And then we fit the sliding window approach, and we fit the standard hidden Markov model, we fit the hidden semi-Markov model on it. And what we ended up seeing was that if you're interested only in what states do people occupy, what states do they go in and out of, you don't care when they do, just what states do they spend time in. All 3 methods perform well. All 3 methods estimate the states very accurately. But if you're interested in the actual state sequences, when somebody switches to a different state, how long they're in that state, then the hidden semi-Markov model far outperforms the standard Markov model and the sliding window approach. Is there a clock in here? Okay. So now I want to switch to showing you an actual application to this. So we applied the hidden semi-Markov model to ADHD research. The findings ended up being pretty interesting. So it is thought that ADHD is driven by dysfunctional connectivity and brain network organization. Right? And here are some of the characteristics of ADHD: hyperactivity, disorganization, inattention, forgetfulness, right? And the collaborators thought, dynamic connectivity might provide unique insight because maybe children or people with ADHD have trouble staying in a certain attentive state before possibly they jump out of it too soon, and that's why they have trouble with inattention or keeping their attention, right? So we had resting state fMRI data. By resting state, I mean you're not performing a task in the scanner, you're just kind of lying there and told not to think about much. So of course, then you think about everything. And they were collected from 38 school-age children with ADHD, and then 79 typically developing children. And we focused our study on 70 regions of interest in the brain. And each of those belonged to one of the following subnetworks that had been shown in the past to be associated with ADHD in static networks. So where just one network was estimated over the course that somebody was in the scanner. So these included the default mode network and some attention networks here, a somatomotor network. And these were the 6 states we ended up estimating, and I'm presenting these in the form of correlation matrices here. So it's kind of hard to see any individual pair here, but just at first glance, you can see that state 6 is very hyperactive, right? There's a lot of red there. There's a lot of positive correlations. You can imagine if you created a binary network here, just tons of edges everywhere. Everything is very highly connected. And then you have some states like state 1, state 3, state 4 that have some negative correlations here. For example, between the default mode network and some of the attention networks. Remember, blue would be a negative correlation. And the collaborators were very excited about this because they said this is what you would expect. Like if someone is thinking about something or focusing on something, that these attention networks would become activated, and apparently the default mode network would become deactivated. And that the default mode network kind of shuts off when some of the attention networks turn on. So they said this is sort of a network, state 1, state 3, state 4, that you would expect if someone's kind of thinking about something or being a little bit more attentive. So we calculated occupancy time for each of these states for the children with ADHD and then the typically developing children. And then we used permutation testing to get p-values here. And what we saw was state 6 was significant. Children spent different amounts of times in state 6, significantly different amounts of times in state 6. And state 3 was as well. This was more borderline significant. And if we adjusted for multiple comparisons, probably here wouldn't be perhaps significant, but still interesting. So state 3 and state 6. State 6 was that hyperactive state. And state 3 was one of those states that had more of the attention qualities here, where the default mode network, we're seeing some anti-correlations between the default mode network and the attention subnetworks. And then we looked at Sojourn distributions here. And I just fit a non-parametric Sojourn distribution. So that's another thing you can do. If you don't want to assume Poisson, if you don't want to assume gamma, you could fit a non-parametric. And sometimes what I'll do is fit non-parametric first. And then if I see it looks like one of the well-known families, then I'll go and I'll fit that particular distribution. But this is just non-parametric for state 3 and then state 6. And you see here, they flip. So state 3, red is the ADHD group. They have a higher probability compared to typically developing of only staying in state 3 for a few short time points before switching out. So this is one of those states that is involved in attention. So what this is saying is the children with ADHD have trouble staying in that for very long before switching to a different state compared to typically developing. And then the opposite is true for state 6, where the typical developing have a higher probability of kind of getting right out of that state once they get into it. And that was that hyperactive state. We also performed a permutation test to see if these differences were meaningful, and we found that they were. I don't know why I don't have the p-values on here, but they were around 0.01, I believe. And then we also looked at transition probabilities. So I kind of created this transition probability map here where the nodes here represent the different states and the arrows represent transitioning to that state. So the thicker the arrow, the higher the probability of going from, like for example, this arrow here is pretty thick. So pretty high probability of going from state 5 to state 6 for the ADHD group. The red arrows indicate where there were significant differences between the two groups, ADHD and typically developing. And so red would mean greater or higher probability compared to typically developing, and blue would mean a lower transition probability in the ADHD group compared to typically developing. It's kind of hard to see the blue arrows here though because they're pretty thin and light. And then here I have the size of each sort of circle here representing each state is proportional to occupancy time in that state. So the ADHD group spent a lot of time in state 6, which is why I have that a bit larger. So you can see here that the ADHD group transition into state 6 a lot, which is making sense because they spend a lot of time there. And particularly, compared to typically developing, they go from state 3 to state 6 quite a bit. So it's like they get into that attentive state, and then all of a sudden, they jump out of it and go to that hyperactive state, more so than typically developing. We can also create binary representations of this state, of the different states. And so what I ended up doing was taking like the top 20% of positive correlations across all the networks, right, across all 6. And I'll just show you for 3 of the states here. Each color represents a different subnetwork. And you can see state 6 has a lot of connections like we thought, because that's the hyperactive one. Another interesting thing, I used iGraph to do this, and I used one of the layouts where it plots nodes that have more connections with each other, right? It plots those closer together, and ones that are further apart in the plot have less connections with each other. So you can kind of see this modular structure emerge here, where the different colors representing those different subnetworks, like default mode network or some of the attentive networks, kind of cluster together, which is really nice because that's showing strong modularity, which represents a more efficient network, right? So nodes that are in the same subnetwork and that perform similar tasks are more highly connected to each other. They're still connected to the other nodes, but more long-range connections. So you can see for states 1 and 3 that nice modular structure emerges where, you know, the blue nodes are clustered together, the yellow nodes are clustered together. Again, these just represent the top 25% of connections. The light blue are clustered together, but then state 6, it kind of becomes a bit more jumbled. So it loses that efficient modular structure in state 6. And again, that was that state that the children with ADHD spent a lot of time in. So some conclusions from this study: children with ADHD spent less total time in and switched more quickly out of states characterized by many negative correlations between the default mode network and the other subnetworks, particularly those attention networks, like the salience ventral attention network. Children with ADHD spent more time in a hyperconnected state, characterized by low modularity as compared to typically developing children. And together, the collaborators were excited and thought that these 2 results imply a mechanism underlying fluctuations in attention commonly observed in children with ADHD. So I'll just end with mentioning an extension I'm currently working on. So a lot of times collaborators are interested in making comparisons, right, between groups of people. Like in this example, children with ADHD versus typically developing, older adults versus younger adults. And so far, we do this by kind of doing some like post hoc analyses where, you know, we fit the model on the whole group so that we get the one big set of, you know, one set of states, and then we go back and we get each subject state sequence, and then we kind of empirically calculate, you know, a transition probability matrix for one group and a transition probability matrix for another group, and the Sojourn distribution for one and the Sojourn distribution for the other, just by sort of tallying up, like, given their state sequence, right? Okay, how many times, consecutive times did they spend in state 1? Save that. And again, save that, and kind of create then the distribution empirically. And then perform permutation testing. But it would be better if we could actually build covariates directly into the hidden semi-Markov model. So we've worked on developing it into the Sojourn distribution aspect of the model, and we've currently done that so that we can kind of directly estimate group effects right in the model for the Sojourn distribution, and then actually test in the model like you would in like linear regression or something. And we eventually want to then do this for the other aspects of the model. But very briefly, for example, if you were fitting a Poisson distribution for your Sojourn distribution, you could do something like this, where the Sojourn distribution for each state K and subject I is now parameterized by the following. So you have this linear combination of effects here. And so then the estimated mean for each state and subject I is given by the following. So the lambda is dependent on that linear combination of effects. And we did some simulations here. We used the SimTB toolbox again, and we simulated for 400 subjects, each with 1,000 time points. We used the same 4 states as in previous simulations that I mentioned. So we just did 10 regions of interest. We kept it simple. We simulated data from some fictitious parameters here. We did 2 binary and 1 continuous. And here were the true values. And here I have the true and the estimated. So we are very close for all of those. So future work will focus on developing covariate implementations for all aspects of the model, not just the Sojourn distribution. Also, developing an implementation that allows for larger network sizes, right? So right now, we're kind of limited because depending on what your sample size is, you're estimating a covariance matrix for each state. And if you have a ton of regions that you're looking at, that gets to be a lot of parameters to estimate. So we're currently working on adding a penalty term here into the hidden semi-Markov model, where we can kind of shrink some of those estimates to 0 and help with estimating the larger network sizes. Also accounting for motion directly in the estimation routine, because we've sort of noticed that sometimes when we fit this model on a group of participants, it's almost like the model detects motion as a state and estimates one of the states to be a high motion state. Like it aligns perfectly with high motion time points. And then obviously to continue to apply these methods to neuroimaging data sets. So right now I'm applying it to a weight loss data set, and we have some interesting findings there, and to an Alzheimer's disease data set. So this is actually joint work with some collaborators from Johns Hopkins University, where I did my postdoc, and some collaborators at UNC Chapel Hill. Any questions?